The overall goal of this technique is to observe two-dimensional mixing of two different surfactants and extract the diffusion coefficient from that.
This technique can help answer key questions in the biophysics and surface science field, such as the fluidity and interactions of different species in cell membranes, as well as interaction between surface active agents. The main advantage of this technique is that we can directly observe mixing of any two surfactant monolayers. Both of them are independently controlled using a very simple setup.
To form a phospholipid monolayer, first prepare a phospholipid solution by cleaning a 4 ml vial with a polytetrafluoroethylene-coated cap. Rinse with acetone, ethanol, and deionized water at least 3 times. Then, blow nitrogen gas thoroughly into the vial to get rid of the water. Dissolve 1 mg of phospholipid into 1 ml of chloroform in the vial to obtain a concentration of 1 mg per milliliter. Next, add dye-tagged phospholipids with less than 1 mol% of the phospholipid solution in order to visualize the phospholipid monolayer with a fluorescence microscope. Wrap the vial with a polytetrafluoroethylene tape to prevent solvent evaporation and store the sample in a freezer at -20°C. To deposit the phospholipids onto the air-water interface, first clean a petri dish with ethanol and deionized water at least 3 times. Then, fill the petri dish with 10 ml of deionized water to create an air-water interface. Spread a few microliters of the phospholipid solution with a